everyone, I am Teacher Yunwei. Welcome to Cancer Unlocked, Understanding Carcinogenic Genes, Part 1. First, we need to understand the origins of cancer. It is related to mutations in oncogenes and tumor suppressor genes. The occurrence of these cancers is positively correlated with our age. In other words, as we age, our risk of cancer significantly increases. Moreover, these gene mutations are cumulative. If a certain level of gene mutations accumulates, it can trigger the cancer process. First, let's introduce what a mutator gene is. Do you remember what a mutator gene is? It's a type of gene that, once mutated, can greatly increase the mutation rate of other genes. We call it a mutator gene. Typically, these mutator genes are involved in DNA repair genes. For example, in the previous unit, we talked about the genes involved in mismatch repair: MUT, MUTEL, MUT-Y. MUT is an abbreviation of the first three letters of mutator. When these mutator genes mutate, the mutation rate of other genes also significantly increases, leading those genes, whether they are oncogenes or tumor suppressor genes, to potentially start the cancer process. It's important to understand that not all gene mutations lead to cancer. Only specific mutations in certain genes will lead to cancer. Thus, when these specific mutations occur, it leads to abnormal cell proliferation and the formation of cancer cells. We call this clonal selection, meaning it has a selective nature. Which genes are mutated to cause cancer? We just mentioned some: oncogenes or tumor suppressor genes. Oncogenes and tumor suppressor genes are involved in DNA repair within the cell, just like the mutator gene mentioned earlier, which is a tumor suppressor gene. Some oncogenes are involved in cell proliferation or in resisting cell death. When these three types of genes mutate, it can lead to cancer. There are 4 steps in cancer formation. The first step is the mutation of genes, as mentioned by the teacher. The second step is cells achieving immortality. Students, are you curious about how long each cell has a lifespan? Cancer cells are different. Cancer cells are immortal. Generally, a cell can divide 50 to 52 times, but cancer cells can continually replicate and proliferate. Once they have replicated and proliferated for a period, it is possible they also accumulate other mutations, leading to cell transformation. After the transformed cells undergo more mutations, they may also develop new blood vessels, spreading through the blood or lymphatic system to other normal organs, causing those organs to also become cancerous. This is called cellular metastasis. This is the 4-step process of cancer formation. First, the teacher will introduce how a cancer cell becomes an immortal cell. This is related to the lifespan of a cell. How can I determine the lifespan of a cell? It is related to the telomeres in the DNA within the cell. We call this telomere. In a somatic cell, the length of the telomeres is fixed. Because telomerase activity is lacking, it is switched off. Every time DNA replicates, it is like a biological clock, each year passing by. The telomeres get shorter and shorter until a certain point when it triggers a stage called crisis or senescence, telling the cell that it needs to quickly undergo aging or cell death. Because DNA without telomere protection is very susceptible to DNA recombination. At this moment, the cell would rather die, age, and die off. This is more stable for the entire genome. Conversely, if this somatic cell undergoes some mutations, activating telomerase again, the cell that should have died ends up surviving. Unstable telomeres can easily lead to gene recombination, resulting in gene mutations and leading to immortality. This is cancer, where gene mutations lead to cellular immortality. Next, let's look at DNA replication causing telomeres to shorten, and when we stop replication, we enter the crisis stage. Typically, immortalized cells cross over this period and constantly proliferate, allowing telomerase to add telomeric sequences to the ends of DNA, thus enabling cells to achieve immortality. Now let's discuss what telomerase is. Telomerase is an RNA-dependent DNA polymerase that adds the sequence TTA-GGG to the ends of DNA. The presence of telomeres stabilizes the entire DNA. It is itself a reverse transcriptase enzyme with its own RNA template that adds a segment of DNA sequence at the ends of DNA. Now, how does a normal cell evolve into a cancer cell? As you all understood, it must start with a gene mutation. Thus, a normal cell goes through gene mutations to become an immortalized cell, then accumulates more gene mutations. As mentioned by the teacher, these gene mutations are cumulative, leading to a transformed cell and then a metastatic cell. On the left side of the slides is the cellular characteristics of a normal cell, and on the right side is a cancer cell's characteristics. We can see that normal cells have a defined lifespan, and they can divide 50 to 52 times, after which they begin to age. However, cancer cells are different. They have an infinite lifespan. Normal cells similar to us require nutrients, serum, and growth factors, but cancer cells do not necessarily need these. They can thrive quite well even without certain growth factors. Additionally, normal cells grow in a flat manner and possess regularity, while cancer cells grow in stacks and are irregular. Thus, if you have a mole or a new spot that is very irregular in shape, you should take special note. Next, normal cells will inhibit growth due to contact between cells. In other words, if cells grow too quickly, they will stop growing. However, cancer cells differ. They lose contact inhibition, meaning even when crowded, they will continue to grow, so they pile up layer after layer again and again. Moreover, normal cells require a substrate to attach to grow. We call this anchorage dependent, but cancer cells are different. They can proliferate even in a suspended state or on soft gels, changing one cell into two cells into 4 cells. We call this anchorage independent, abbreviated as AI. Furthermore, if we inject normal cells into an immunocompromised mouse, they will not form tumors. But if we inject cancer cells into nude mice, they can form tumors. This is the difference between normal cells and cancer cells. Next, the teacher will introduce HeLa cells. HeLa cells are the first human cells that can be cultured outside of the body. They are cervical cancer cells. For many years, these cells, due to their immortalization characteristics, have been continuously reproduced globally, exceeding 5,000 tons. I believe it has surpassed that by now, weighing as much as 100 Empire State Buildings. Later, an American author and biomedical journalist published a book titled The Immortal Life of Henrietta Lacks. So who is Henrietta Lacks? We will introduce her shortly. She was a young woman from a small town in Virginia, engaged in tobacco farming, living in poverty. In 1951, at just 31 years old, she had 5 children and was diagnosed with cervical cancer at Johns Hopkins Hospital, where it was discovered that she passed away 8 months later from this cancer. Moreover, doctors found that the cells cultured from her tissues had rare proliferative abilities. Thus, this scientist named the cells by combining her first and last name, creating HeLa cells. This is how HeLa cells originated. This was the first human cell line to be cultured outside the body, and they were stored in the American Type Culture Collection, abbreviated as ATCC. Later, scientists acquired this cell from ATCC, continuously transferring and culturing it, considered it an immortal, endless cell line. These cells were used for research on polio, smallpox vaccines, viruses, in vitro fertilization, the cloning of sheep, and DNA mapping, all thanks to the involvement of HeLa cells. Next, the question arises. HeLa cells were preserved and continuously reproduced, even sold to various medical centers, but at that time, her family was unaware. It wasn't until 20 years after Henrietta Lacks passed away that scientists, wanting to investigate why her cells were immortal, sought her husband and children for research, which led to the exposure of HeLa cells. At that time, the rights of Black individuals, especially poor ones, were neglected. Thus, scientists in the biomedical field also held an attitude of intellectual arrogance, never considering that today in our society, we emphasize intellectual property rights very highly, which involves the rights concerning human-derived materials, including organs, saliva, and cell rights. At that time, due to a lack of understanding and awareness about rights, her husband was very angry and even complained, saying that they were profiting off the sale of my wife's cells while they took advantage of her. That issue had persisted for 20 years. For a rural Black farmer at the time, he did not know how to fight for his rights. Now the teacher wants to ask you all, do the following characteristics belong to normal cells or cancer cells? Needs attachment to grow, needs serum, grows in a single layer, inhibits growth due to contact? These belong to normal cells. Next, which of the following physiological features are characteristic of cancerous cells? 1. Will not form tumors in nude mice. 2. Has a defined lifespan. 3. Does not need growth factors. 4. Cells can form colonies on soft gels. 5. Will form tumors in nude mice. 6. Grows in stacks and irregularly. The answer is 4. Can form colonies on soft gels, will form tumors in nude mice, and grows in stacks irregularly. Did you answer correctly? Next, the teacher will introduce 2 terms. The first is primary cells. What are primary cells? They are cells that can be cultured directly from animal organs, such as the liver or lungs. We call them primary cells. After a period of culturing, for example, after they have been replicated 50 or 52 times, these cells will naturally die off. If primary cells break through the crisis, accumulating some gene mutations, we call them established cells. Typically, established cells have some chromosomal variations. Normal cells exposed to carcinogenic substances will lead to gene mutations and transformation. Transformed cells under a microscope will appear as a focus, and their genomes will exhibit aneuploidy, meaning chromosomes may be duplicated or deleted. During the process of cancer formation, the first step is always to cross over this crisis period to become immortal cells. Immortal cells at this point still need to attach and may need nutrients to grow. Once they accumulate further mutations, they may not need nutrients to continue growing, and their shape may change into a focus-like appearance, leading to what we call transformation. Cancer cells can spread through the blood or lymphatic system to other normal organs, which we refer to as metastasis. Primary cells are directly cultured from organs or tissues, so they are closer to the actual state. Established cells, or immortal cells, are those that have gone through the crisis stage and may have some gene mutations. This diagram shows a soft gel. We separately planted cells on this soft gel. If cells can grow, we call this anchorage independent. If they cannot grow on the soft gel, we call this anchorage dependent. Do you remember? Cells that can grow on the soft gel we abbreviate as AI, which belongs to cancer cells. The cells above can proliferate on smooth soft gels, forming a colony. Normal cells cannot form growth on soft gels. We call them anchorage dependent, as they require attachment to grow. Next, the teacher will quiz you again. Which are cancer cell characteristics? 1. Needs attachment. 2. Needs serum. 3. Provides growth factors. 4. Grows in a single layer, flat. 5. Will form tumors in nude mice. 6. Does not need attachment. 7. Can survive even without serum. 8. Grows in stacks and has rounded cells. 9. Exhibits anchorage-independent phenomena. The answers are 4, 5, 6, 7, and 8. They will form tumors in nude mice, do not require attachment, can survive without serum, grow in stacks and have rounded cells, and can form colonies on smooth soft gels. We call this anchorage independent. After understanding the differences between normal cells and cancer cells, students, you can see that cancer cells do not need to attach, and even without serum, they can grow, they can stack up, and their cells are rounded. In contrast, normal cells grow in a single layer and are flat, and they require attachment to grow. Now the teacher will discuss the relationship between cancer cells and the immune system. Cancer cells, in addition to constantly proliferating, it can even transfer to other organs or tissues. It also possesses a capability to deceive and suppress the immune system's detection and elimination abilities. Cancer cells use the immune system to fight against it. Scientists have identified 4 ways, as shown in the slides. Firstly, it can secrete cytokines that inhibit immune cell function, such as TGF-β. On the surface of cancer cells, proteins like PD-L1 are expressed that can suppress immune cell functions. This is akin to whether anyone has seen the invisibility cloak in Harry Potter. These cancer cells are quite special. When immune cells attempt to attack them, they don this invisibility cloak, preventing immune cells from detecting them. Moreover, they make immune cells insensitive to the cell death stimuli induced by T cells. They secrete lactic acid, causing the tumor microenvironment to be more acidic, which is unfavorable for T cell activation. Furthermore, recent discoveries by scientists reveal that these cancer cells have developed a way to counteract the immune system. Dying cancer cells try everything to block the immune system from attacking the surviving cells. Dying cells will pump out a lot of potassium ions, making the potassium ion concentration in the tumor microenvironment particularly high. When potassium levels rise and these ions enter T cells, it affects their ability to secrete interferon G. This means that the weapons T cells use against cancer cells are reduced. When excessive potassium ions enter T cells, they inhibit a signaling pathway known as AKT-mTOR. AKT-mTOR primarily activates T cell metabolism. High potassium levels blunt T cell function. In other words, even though this cancer cell dies, it still tries every way to ensure that the surviving cancer cells can survive robustly. After scientists understood this method, they introduced a pump into our T cells through genetic engineering. This pump, known as KCNA3, successfully pumps potassium ions out of our cells, reducing their concentration in T cells. This means that the suppressed T cell function can be restored. The imbalance in the tumor microenvironment's concentration also hinders the immune system from executing its task of clearing cancer cells. Today, our take-home message is that the origin of cancer arises from mutations in oncogenes and tumor suppressor genes. When these oncogenes and tumor suppressor genes mutate, they initiate a 4-step process that starts cancer. The first step is gene mutation, followed by cellular immortality, cellular transformation, and even metastasis. A cancerous cell has no lifespan. When mutants reach a certain extent, they do not need to provide growth factors to form colonies on soft agar, generating tumors in nude mice. They grow piled and irregularly. Do you all understand these take-home messages?